Today, I'm going to be talking about the impact of fats on the gut microbiome. My research question was, how do different types of fats impact the composition of the gut microbiome and the development of metabolic disorder? For a quick introduction to common dietary fats, all fat-containing foods often consists of a mix of these three fats. The carbon skeleton of a saturated fat is completely saturated with hydrogen, creating a structure without any bends. This type of fat is a solid at room temperature. Monounsaturated fat has one double bond between two carbons present in the structure, creating a bend. This type of fat is often a thick liquid at room temperature. Polyunsaturated fat contains two or more double bonds within its structure, creating multiple bends. It is a liquid at room temperature. Omega-3 and omega-6 fatty acids are both a type of polyunsaturated fatty acid. Two of these fats are considered essential in the diet, meaning that since the human body cannot synthesize them endogenously, they must be consumed from food. Alpha-linoleic acid, one of these essential acids, is an omega-3 that functions to lower cholesterol and blood pressure, decrease inflammation and blood clotting, stimulate vasodilation, and is crucial for brain function and human development. It is converted into other omega-3s, such as EPA and DHA, as well as eicosanoids, which are lipid-like metabolisms crucial for the immune system. Linolenic acid, the other essential fatty acid, is an omega-6 that also promotes brain function, growth, and development. However, overconsumption can lead to increased inflammation and blood vessel constriction. Linolenic acid is converted into other omega-6s, such as arachidonic acid, as well as eicosanoids. The ratio of dietary fats, saturated versus unsaturated, as well as the ratio of omega-3s to omega-6s consumed, is strongly correlated with the development of certain disorders, such as cardiovascular disease, inflammation, and metabolic disorder. When talking about the gut microbiome, we are referring to the countless number of non-pathogenic bacteria that exist in the gastrointestinal tract. These bacteria aid in digestion, produce short-chain fatty acids, and synthesize vitamin K and many B vitamins. The specific type of diet consumed by an individual has a distinct impact on the composition of intestinal bacteria. Current research has shown a correlation between diets high in fat, especially saturated or omega-6 fatty acids, and negative health outcomes such as obesity and inflammation. The microbiome plays an incredibly important role in the development of these health outcomes, with the proliferation of certain bacterial strains impacting processes such as lipid deposition, gut permeability, inflammation, and insulin sensitivity. Fecal studies of obese versus normal weight individuals have shown a significant difference in the composition and abundance of certain bacterial strains in the GI tract. During trials performed on laboratory mice fed a high saturated fat diet, the concentration of hydrogen sulfide producing bacteria increased significantly over time, while bacterial levels stayed the same in mice fed with omega-6 polyunsaturated fats, and levels decreased in mice fed an omega-3 based diet. The bilophila and dissolvovibrio greatly increased, as well as erysipelas trichiaceae. The population of Enterococcus and Lactospiraceae increased as well in the mice fed a high saturated fat diet. Meanwhile, in mice fed a high polyunsaturated and monounsaturated fat diet, the level of dissolvovibrio was low, while populations of Sotorelliaceae, Merospirillum, and Mucilaginibacter dagianensis were higher. On trials performed using human subjects fed different fat-enriched diets, it was found that after consuming a diet rich in mono and polyunsaturated fats, the lactic acid bacteria population greatly increased. Lactic acid bacteria perform multiple beneficial functions, producing lactic acid, which protects the gut pH, antimicrobial peptides, which are part of the immune system and acts as a probiotic. In subjects fed a lower fat diet in general, the microbial alpha diversity increased, as well as Faecalibacterium and Blaudia population. Alpha diversity is a measure of how many different bacterial strains are found within a single sample, as well as their abundance. Faecalibacterium produces butyrate, which has anti-inflammatory properties and protects the gut barrier, while Blaudia has also probiotic properties. Compared with the lower fat group, the higher fat group significantly increased the fecal concentration of palmitic acid, stearic acid, arachidonic acid, indoleacetic acid, indole, and picresol. Elastipase, while sometimes having anti-inflammatory effects, is also linked to neurological conditions, colorectal cancer, and inflammatory diseases. Similarly, a decrease in the amount of short-chain fatty acids can lead to inflammatory bowel disease, metabolic disorders, cancers, and impaired brain and immune function. Short-chain fatty acids are also essential to insulin sensitivity and a healthy GI tract lining. In each box plot, a percentage distribution as well as the significant difference of bacterial population is shown for 8 different bacterial families found in the gut microbiome of mice. In this clinical trial, 3 groups of mice were fed different diets, one enriched with monounsaturated and polyunsaturated fats, one enriched with saturated fats, and one fed a standard chow diet labeled SD. These plots show how the consumption of different fats leads to the proliferation of significantly different microbial families. Changes in the gut bacterial population directly correlate with the development of cardiovascular and metabolic diseases. Certain types of bacteria, when growing overpopulated, can harm the makeup of the gut microbiome and cause a host of harmful effects on the body. Regarding metabolic disorder and cardiovascular disease risk, both normal weight and obese individuals fed diets high in mono and polyunsaturated fats saw a decrease in BMI, while diets high in saturated fat and omega-6 fatty acids resulted in more weight gain compared to the control group and those consuming high omega-3 based diets. Compared to the control, high saturated fat and omega-6 diets had a more mesenteric, perigonadal, perineal, and subcutaneous fat deposition. Meanwhile, the control groups and the groups fed omega-3 based diets were similar in measures of adiposity and lower than the groups fed saturated fat and omega-6 fatty acids. Regarding the impact of specific bacteria, dissolved Vibrio, as well as hydrogen sulfide-producing bacteria such as Bilophila, contribute to inflammatory bowel disease and colorectal cancer. These bacteria thrive in bile-filled environments, making diets high in animal fat a cause for their proliferation. Dissolved Vibrio is also associated with increased plasmatic insulin, systolic blood pressure, chronic liver disease and cirrhosis, obesity, and diuresis, which is increased urine production. Enterococcus and Lactospiraceae, also increased after diets high in saturated fat, are both linked to the development of obesity and diabetes, and have been shown to proliferate when the gut microbiome is in dysbiosis, which means it is imbalanced. Allistipes and Erysipelotrichaceae, abundant after a high saturated fat diet, are linked to inflammatory bowel disease and colon cancer. In addition, Allistipes is also associated with increased plasma insulin levels and the development of neurological conditions. Short-chain fatty acid production is essential for maintaining the lining of the gastrointestinal tract and insulin sensitivity. This production is decreased in high-fat diets, which can lead to inflammatory bowel disease, cancers, metabolic disorders such as obesity and diabetes, and impaired brain and immune function. However, lactic acid bacteria, the levels of which are greatly increased in diets high in mono and polyunsaturated fats, perform multiple beneficial functions, producing lactic acid, antimicrobial peptides, and act as a probiotic. Lastly, it was observed that individuals consuming a low-fat diet had decreased levels of the metabolites indole and picresol, while higher-fat diets led to an increase of both. Indole is produced in the gut during the breakdown of the amino acid tryptophan and is linked with hypertension and cardiovascular disease. Picresol, produced in the gut during the breakdown of the amino acid tyrosine, is linked with chronic kidney disease and diabetes mellitus. This figure comes from an animal study where mice were fed a diet high either in saturated, omega-6 polyunsaturated, or omega-3 polyunsaturated fats. After 2 months of feeding, the change in weight, body fat percentage, and HOMA-IR can clearly be seen throughout the different groups. Despite being fed the same amount of calories, as well as the same caloric percentage of fat, mice fed a high saturated fat and a high omega-6 diet gained significantly more weight and body fat percentage. In addition, both had high HOMA-IRs, especially the saturated fat group. HOMA-IR is used to estimate insulin resistance, with higher HOMA-IRs indicating increased insulin resistance, which is part of the progression of type 2 diabetes mellitus. In the group fed a high omega-3 diet, HOMA-IR decreased, indicating increased insulin sensitivity. In conclusion, in order to maintain a healthy functioning gut microbiome, we should try to consume a diet with a specific fat profile. As studies have shown, diets high in fats but with a good ratio of different fats are able to maintain normal gut function and limit adverse health effects. Prioritizing fat intake from mono and polyunsaturated sources, limiting processed food intake, and avoiding overconsumption of animal products ensures that our microbiome functions in a beneficial way and reduces the risk of developing metabolic syndrome. Some recommended sources of fats include avocados, nuts and seeds such as chia seed, flaxseed, and walnuts, fish including salmon, tuna, sardines, and mackerel, and plant oils like olive oil, canola oil, and sunflower oil. We are what we eat. Our bacterial population depends on what we consume, and our overall bodily health depends on that bacterial makeup.